In this video, I will show you the spread plate method. Spread plate method is a dilution method that helps you to answer the question, how many bacterial cells do you have per ml of a sample or per gram of a sample? The sample that I have chosen for you is soil. If the sample was a liquid sample, I would have started with 1 ml of it. This is a solid sample, so I wait for you 1 gram of soil, and I will transfer them to my first test tube, which contains 9 ml sterile saline. Because the sample is soil, so I will label this test tube. This is my first dilution, 10 to the power minus 1. Because it's soil and it's not gonna dissolve inside the liquid, I need to vortex it in order to mix properly the bacteria inside the liquid that I have. This instrument is called vortex. You press on it, it mixes the content of the test tube. Then I label the rest of my test tubes, 10 to the power minus 2, minus 3, and minus 4. Each one of them contains 9 ml sterile saline. Then I get a pipette. This pipette can measure between 100 and 1000 ml. In order to continue the serial dilution, I needed to transfer from one tube to another 1000 ml, which is 1 ml. So I adjust my pipette until I read 1000 microliter, which is 1 ml. I can read now 1000. Sterile tips, and I take one tip and I transfer from the first test tube 1 ml to the second test tube. When we are performing serial dilution, we need to change the tip, throw it in the hazardous waste after each step. Even the second test tube, it's better if you vortex it. I transfer from the second to the third. No need for vortexing here, simple mixing is enough. And then from the third to the fourth. I prepare as many test tubes as I want, depends on my experiment. So this is 1 to 10 serial dilution, and it is done. I will continue with the process. I will take I still need the tips. I will take 4 Petri dishes. I will put them in front of me. And I need to label them. I label each one of them with the name, the group number, and the dilution factor. This is 10 to the power minus 1. Again, name, group number, 10 to the power minus 2. Name, group number, 10 to the power minus 3. And then name, group number, 10 to the power minus 4. Again, I remind you, I don't label on the lids, I label on the side of the agar itself. I change the number on my pipette to make it 100 microliter. The spreaded the plated amount is 100 microliter. If I want to save resources, I can start from the most diluted samples to the least diluted, means from 10 to the power minus 4 to 10 to the power minus 3, and keep using the same tip and the same spreader later on. So I'm transferring 100 microliter from each test tube to the appropriate labeled plate. When you are pipetting, feel the tip of the pipette when you press. There are 2 sides of your pressing. You can press gently or you can overpress. When you fill, press gently, suck the liquid, and when you empty, overpress. So if you overpress and you suck, you will take much more than the number written. So press gently, empty the air, take the amount of liquid necessary, and then overpress to empty. In this step, you need to be fast. We want to spread the liquid on the Petri dishes. I will take a sterile plastic spreader. In research labs, they use glass spreaders that are reusable. You can sterilize them with with heat and alcohol. So I will take my first Petri dish. I am turning the Petri dish in my hand clockwise, anti-clockwise, and the spreader on the Petri dish clockwise. I keep on doing this until I see the liquid in front of me totally distributed and dry on the Petri dish. This is my first Petri dish. Keep it upside down. Second one, I'm always trying to do this process next to the heat, whether it's a flame or an incinerator. Again, why am I using the same spreader? Because I am moving from the most diluted sample to the least diluted sample. I am moving from 10 to the power minus 4 to 10 to the power minus 3 to 10 to the power minus 2, and then the last one is going to be 10 to the power minus 1. So the last Petri dish, I am next to the heat at all time. When I am done, this spreader is not reusable. I have to discard it. So I will discard it in the hazardous waste. My Petri dishes has to go upside down in the incubator. Each colony will grow by the next day. Each bacteria will grow by the next day into a colony. The way the cereal dilution plates will look like is this way. This is a 10 to the power minus 1 Petri dish. This is a 10 to the power minus 1 Petri dish. And then this is a 10 to the power minus 2 Petri dish. And then this is a 10 to the power minus 3 Petri dishes. If you try to count the number of colonies, you will see that I have more colonies here, and then less, and then less. To calculate the CFU, here I have one colony only. To calculate the CFU, you pick a plate that has between 25 and 250 colonies. In this case, your first Petri dish is appropriate for calculation of CFU. You count the colonies, and the CFU will be the number of colonies divided by 0.1 and divided by the dilution factor. This is also a cereal dilution of soil. This is 10 to the power minus 1, very thick. Minus 2, still very thick. Minus 3, the number of bacteria start becoming visible. And here the minus 4 is countable. I wanted to show you this to show you that in the soil we have different types of bacteria. You can identify this yellow large colony, this medium orange colony, this red or brown medium in size colony. You need to count to calculate the total CFU. You need to count all of them and divide this number on the plated amount times the dilution. That's it for the spread plate method. Hope it's clear.